Hello, my name is Dan Isom. I'm an assistant professor of molecular and cellular pharmacology at the University of Miami Miller School of Medicine. I'm here today to talk about CRISPR-based technologies for exploring the dark GPCRome. The work I'm going to talk about today was done by three very talented graduate students, Nick Kapolka, Jeff Tagan, and Jacob Rowe. In the lab, we use a multidimensional approach for GPCRs and beyond. We use yeast for discovery and for profiling of GPCR ligand interactions. We use in vitro cell models for validation of our yeast discoveries. Typically, we use HEK293 T cells and Brett-based GPCR assays. We also do a lot of informatics and computing and synthetic biology. Together, these approaches form the backbone of much of the profiling assays we develop. Although GPCR signaling needs no introduction in this audience, I included this slide for one very specific point, and that is that when you do activate a GPCR, you activate a G-alpha subunit. And this G-alpha subunit is a G-alpha subunit from one of the four families, S, I, Q, and 12. And the assay that I'm going to discuss, we always interrogate all possible GPCR G-alpha combinations. So I just want you to keep this in mind. So in the lab, we use both yeast and HEK models primarily. In our yeast model, we use Saccharomyces cerevisiae, which has only one GPCR pathway. We've already published a few papers on assays we have developed for GPCRs in this system. In case you're interested, I'm going to describe two of those technologies today. When we do make a discovery in yeast, the first thing we do is we try to validate it in a HEK system using either a mini G protein assay out of the Lambert lab or two assays out of the Roth lab associated with the IDG, either the TruePath or the Presto Tango assay. So it's nice to have a number of different assays with a number of different readouts to validate our findings. I would also like to point out that our technologies draw heavily from the IDG resources provided by the Roth lab, mainly access to receptors and receptor sequences. So we're very thankful for that. Today I want to talk about two high throughput CRISPR technologies in yeast. They're related. One we call DCYFIR, which stands for dynamic cyan induction by a functional integrated receptor. And two, I want to talk about a multi-pedded version of DCYFIR. A few key points of emphasis are that, number one, these approaches are low touch. They're driven by intensive computational and robotic automation. They're high throughput. We have generated many thousands of yeast strains even without even trying. And they're very collaborative. Our applications can target pretty much any drug target, including dark kinases and ion channels, anything for which we have a functional readout. So they're very powerful and they're very broad. One of the reasons we like using yeast so much for profiling is that they're very genetically malleable and that CRISPR is very efficient in yeast. And this is because if you make a double-stranded break in the yeast genome, they fix it almost exclusively by homologous recombination, whereas in our cells, we use non-homologous end joining. As a result, what we can do is we can co-transform yeast plasmid containing the necessary CRISPR machinery, along with a linear piece of DNA that has the DNA payload, whatever you want to deliver, flanked by homology to the yeast genome. So when we do this transformation, we get colonies on the plate, and in our hands, in practice, 90% of these colonies have the intended genome edit. So this is very efficient, and it can be different types of edits. It can be deletes, it can be knock-ins, it can be anything you can really imagine wanting to do with the genome. So what we did was we took these CRISPR capabilities and we began to humanize the yeast model. And to review, Saccharomyces cerevisiae has one GPCR pathway known as the pheromone pathway, so there's essentially no crosstalk. And what we did was we deleted two negative regulators of the pathway, a GAP and a cell cycle arrest factor. This made sure that the cells grew even after we treated them with ligand. We also installed, we replaced a known pheromone responsive gene with a transcriptional reporter, the cyan fluorescent protein M turquoise 2. So now when these cells are activated, they will glow cyan. In the second step, we removed the endogenous receptor to make way for human GPCRs. Before installing human GPCRs, though, what we had to do is we had to install a CRISPR-addressable landing pad, and we have installed several of these, and they all look the same to date. They are comprised of three components: a constitutive TEF promoter, a SIC terminator, and a unique targeting sequence. In this case, it's the 10-2 targeting sequence. The 10-2 indicates the genome locus where we have installed the pad at a safe harbor site in the yeast genome. So once we had the padded strain, what we did was we created 10 versions of the padded strain that covered all possible GPCR G alpha signaling combinations. And to do that, what we did was we took the yeast G alpha subunit, and using CRISPR, we just edited the last 5 residues to swap in 5 residues from one of the human G alpha subunits. And due to sequence degeneracy in the last 5 residues, it only takes 10 of these chimeric, C-terminal chimeric G alpha subunits to cover all possible GPCR G alpha coupling combinations in yeast. So when you put this all together, what you get is a yeast strain that expresses a human GPCR from the genome from a genetically integrated landing pad, a CRISPR integrated landing pad. This human GPCR couples to one of the 10 G alpha chimeras in its particular strain, and stimulation of this pathway results in cyan cells by driving the M turquoise 2 transcriptional reporter. So these cells, when they're on, are going to be cyan. So when we do these experiments, though, we always install a given receptor into all 10 strains to interrogate all possible GPCR G alpha combinations. And an example of that is shown here in representative data for the arginine vasopressin receptor 2 and the melatonin receptor. In these plots, the white bar is the untreated condition and the red bar is always the treated condition. In the case of AVPR2, it's very specific. It only signals in the presence of agonist in the S strain. But in the case of the melatonin receptor, we see signaling through all 4 of the I strains, and the signaling is pretty much the same intensity. So this is a very strong GI coupled receptor. So with this capability in hand, we then took our first big technological step in terms of innovation, and that was we began building a pipeline that enabled us to install many human GPCRs into yeast using the CRISPR strategy, transformation strategy I mentioned a few slides ago, using that to install GPCRs into all 10 strains, screening those strains, and essentially building up a library of yeast strains that contain human GPCRs coupled to specific G alpha subunits. So we did that for 30 receptors. And so for each receptor, what we needed to do was validate that it responded to its known ligand. And so in this case, the adenosine receptor ADORA1, yes, responds to adenosine. And we did this for all 30 receptors. So, we have at least 300 results. We have 300 strains. In some cases, we did more than one ligand, but in most cases, we did just one endogenous ligand. And the main take home here is all of the signaling you see with all these bars really recapitulates what's known in humans. So there's very good agreement between the yeast system and the human system. The exception here is that we can parse out contributions from the different G alpha subunits, which you could not do in a HEK cell. So there are pros and cons for both approaches. It's best if you combine them in our experience. So of these 30 receptors, 5 are in the dark set of GPCRs, which is why we're here today. This is what we're interested in. GPR4, GPR65, and 68 are the proton sensing receptors. This is hydroxycarboxylic acid receptor 3. I'm going to talk more about that today, mostly about that today, and the succinate receptor, which I'm not going to talk about. So now we sit with 30 GPCRs, 300 barcoded strains. The strains are barcoded with the receptors. They're barcoded with the G alpha subunits. many of you can now imagine the next technical step we took to innovate this system. And that is, we took all the strains together from the freezer and combined them together into a plex, into a multiplex experiment, and we call this DCYFIRplex. And this is how this is done. We combine all of the strains and we add a ligand. This ligand can be an endogenous ligand from the set. It can be from a library. We've screened libraries. We've screened endogenous ligands. We let the cells incubate with the ligand for some period of time to allow the GPCR to be activated. We then add a red tracer, which is a yeast strain that is just expressing red fluorescent protein. And the reason we add that red tracer is that we then go to the cell sorter and we use that red tracer to count sorting events between experiments, and we always collect the same number of cells. What cells are we collecting? We're collecting all those cells into an active pool that turns cyan. So any yeast strain that turns cyan, we know is activated by the given ligand in the pool. And so we collect that active pool. Of course, we don't know all of the receptors that are in that pool. We just know that they are yeast cells that turn cyan. So in practice, this is what this looks like under the confocal. We have 3 representative examples of the 300-plex treated with SRIF14, which is a peptide agonist for the somatostatin receptor 5, melatonin, which is an agonist for the melatonin receptors, and sphingosine 1-phosphate, which is an agonist for the sphingosine 1-phosphate receptors. And so in all 3 cases, you can see a red tracer, and you can see cells that are cyan, so they've been activated. But when you look at the merge, you can see in the plex that there are many yeast strains that are not activated. And this is just the nature of the plex. Only a subset of the cells are going to be activated. So of course, using gating, we can sort out the tracer population, the inactive population, and of course, we can collect the active pool. And that's what we do. We collect the active pool and we deconvolute what's in that active pool by quantitative PCR on the genomic DNA. So we do a simple genomic DNA prep. As a reminder, on the x-axis here, we have 30 pooled GPCRs that correspond to 300 Decipher strains. So when we deconvolute this with the qPCR, we only get signal from somatostatin receptor 5, and that signal is 2 to the 7th. So this is a massive signal to noise. Likewise, when we treat with melatonin, we pull back the 2 melatonin receptors. When we treat with sphingosine 1-phosphate, we pull back the sphingosine 1-phosphate receptors. And I'd like to point out that this process is very highly automated. Computationally, the primers were designed. It's tricky, but we know what we're doing, and it's very slick and robust. And all of this is handled by liquid handling robotics. So it's really high throughput, a lot of repeats, N is high. It's a really nice method. Of course, we had to validate this against all of our known GPCR ligand interactions, and that's what this slide just conveys, that basically we did the testing, we did the plexing, all the receptors responded to their known agonists. We also took an orthogonal approach just for proof of concept where we deconvoluted using nanostring, which instead of looking at the genomic DNA, looks at the mRNA population. On the y-axis here, we have the different agonists. On the x-axis, we have the different receptors. And this dark purple line you see here is just telling you, basically, you get what you expect, that those endogenous agonists that are supposed to activate their given receptors are. So this was great confirmation that regardless, independent of the readout, you were getting the same result. So you can imagine that when you mix a whole bunch of GPCRs together into a single tube, 300 to be in fact, 300 in the case here, that you find unexpected things. And this is what we're after in the first place, and making these libraries as big as they can be so we can do this sort of discovery. And so, you know, we have screened the plex against metabolite libraries. We have screened the plex against other things. But what I want to focus on today is our very first discovery, where we just screened the plex against a metabolite and made a pretty neat finding. So we screened the plex against this tryptophan metabolite, kynurenic acid, which is very important, which is a very important tryptophan metabolite and is important for inflammation. It was a known agonist for GPR35, but we found that it was also an agonist for HCAR3. It also turns out to be an inverse agonist for ADRA2B, an adrenergic receptor, but I'm not going to talk about that today. So what we did was we identified that this kynurenic HCAR3 interaction is coupled through GI. And then, as I said earlier, we went from yeast to the HEC system and validated that this interaction is real in a human cell. And in fact, kynurenic acid is more potent than its known interaction with GPR35. So we know this is real and we think it's important. But why is it important? Why is it new? It's amazing to go from the yeast to the HEC system. And we think it's important for this reason. It was already known that HCAR3 was agonized by a metabolite. It was agonized by a metabolite from the beta oxidation pathway, 3-OH octamate, which is right here. And in that system, 3-OH agonism of HCAR3 is actually anti-lipotic, meaning it stops lipolysis. Likewise, it appears that kynurenic acid may agonize HCAR3 expressed on immune cells, mainly neutrophils and macrophages, to be anti-inflammatory, to combat inflammation. So this is probably a pretty important drug target and a pretty important target for understanding biology. But let's take a step back and understand why these two functions kind of mirror each other. So if you go to the fat cell, what you'll see is that when you're fasting, hormonal signals activate the beta-adrenergic receptor, which activates adenylate cyclase and causes lipolysis and free fatty acids to form. These get beta-oxidized, but when you form too much of this intermediate, it agonizes HCAR3 as a negative feedback to inhibit adenylate cyclase so you don't waste energy, right? So 3-OH actually negatively feeds back to regulate the adipocyte from wasting too much energy. Likewise, we think kynurenic acid, when it's made in an inflammatory response, negatively feedbacks on immune cells to keep them from overacting. So we think this is a very interesting parallel in biological function that is ready for exploration. So yes, we've done it. It's very interesting. We were able to make a discovery from profiling yeast to HEC, and we think it has new biological insights. So this is very fulfilling. And so to close out the talk, what I want to do is I actually want to focus on another approach, and that is the multi-padded decipher, just really quickly. And so what we did was we took our single padded strains and we just added 3 more CRISPR-addressable pads at 3 other genome locations. This is pretty straightforward. They all have the same architecture, they all have the same promoters and same terminators. In our first application, what we did was we took a dark receptor, GPR68, and as you see, when there's 1 copy of GPR68, there's no signaling at Q, 2 a little bit more, 3 a little bit more, and 4 a little bit more. Why am I picking on Q? Well, GPR68 is known to couple to G-alpha Q. So the fact that it wasn't coupling to G-alpha Q when only 1 copy was present, and it was coupling when more than 1 copy of GPR68 was present, was just showing us that we were titrating this interaction here by including more copies and utilizing those 4 pads. Likewise, we also did a genetically encoded experiment where in 1 pad we made a GPCR, which was the somatostatin receptor, and in the other we made its peptide agonist. And sure enough, when you express the peptide agonist from the genome, and we also put a little bit on it so it's stuck inside the cell wall, you agonize the somatostatin receptor and get back the response that you would get if you had added exogenous peptide. So that brings to a close those 2 applications, and I just wanted to bring your attention to some upcoming papers that also involve dark GPCRs. We have one on the molecular evolution and mechanisms of human GPCR pH sensing, and we have another one on the pH regulation of human GPCR signaling, and I can talk more about that in the open session if you like. So to summarize, we have a suite of high-throughput CRISPR technologies applicable to GPCRs, to other dark targets, and we do have other emerging technologies in the CRISPR world. By combining yeast profiling with in vitro HEC validation, we discovered many new GPCR ligand interactions. I told you about one today, HCAR3, and that we think it's important for the immunomodulatory functions. And lastly, I'd like to thank the organizers and the IDG for inviting me to give this talk and for their support. Thank you.